So, usually, I want to make sure that I have time to go to church in the morning, and then we have our study sessions, and then our families eat lunch together. So, let's say around 2 o'clock. Does that work? 2:30 gets better, or 2:30?
Yeah.
Okay, 2:30 on Sunday. So, yeah, so what we'll do is we'll have it in the E building where you have your lab. There's a lecture room there, E6, we'll meet in there. And here's how study sessions work, is you need to show up with some questions, and then I help go over stuff that you did together you're not sure about and all that kind of stuff. And then if we have time left over, then what I do is just kind of throw out questions, and you can try to answer them and kind of see what you know and that kind of stuff. And if it's towards the end of the time, where we're at, it's absolutely no different. But you might do 2 days. You may not be filming any questions like that? All right. You have my home phone number, right? Okay. My biggest fear is I'm going to have a study session and then forget and not show up for it. And so I haven't done that yet. But I'm also, a lot going on. It's going to happen someday. If it's 2:30 and I'm not here or in my office, look in my office first. Knock on the window if I'm sitting there. But if it's 2:30 and I'm not here, call my house and tell me where I'm supposed to be. And tell me if I'm supposed to be to school. Oh yeah, you guys can email me anytime you want. So yeah, you email me questions. If you're on campus, just drop by my office. We'll go over stuff. So yeah, no, absolutely not. All right. So when we left off last time, we actually got past this point, but real quick, just a review. So the cutaneous membrane is basically kind of what we think of as this outer portion. We talked about our integumentary system. It's all of this. So the cutaneous membrane is the epidermis and the dermis. And then we also, when we talk about the integumentary system, we're also talking about the hypodermis. So we have 3 basic components that are here. When we take a look at the dermis, we have a papillary region and a reticular region. We'll talk about how we kind of decide which part is which in just a little bit. And then we have all these other parts that are associated with it. We have oil glands and we have sweat glands. So the oil glands are sebaceous glands, the sweat glands are sudoryphrous glands. We have hair that's associated with it. We have all these tactile structures that are present, and then obviously vascularization and those kinds of things that occur in this particular way. And then when we take a look at our skin, we said we had 2 types. We had thick skin, we had thin skin. So where do we find thick skin? The palm, sole. Everywhere else is skin. Thick skin typically has how many layers? 5. And thin? 4. Okay. And when we take a look at these guys, and we talk about thick skin, when we talk about the layers, one of the things we'll notice is like this outermost layer is generally significantly thicker on thick skin than it is on thin skin. And so if you look at your hands, you've got places in your hands where this is thick, and then you've got other places in your hands where it's a whole lot thicker. So there are calluses and things that are there. And so our skin responds to stress, and that outer layer is stratifornia, and it's what's sloughing off all the time. And so I mentioned before that I think we were talking about the cell connections and how the feels tissues held together. And if these guys are pretty tightly held together, and the connections are desmosome. And so that means the cells fall off individual kinds of cells. And then the next thing we talked about, we were taking a look at that bottom layer. That's called the stratabasal. And the cells that are called the germinal cells or basal cells. So we're going to take a look at several different kinds of cells that we're going to find in epidermis. And the role of this guy was to produce more. So go through mitosis or reproduce, right? High ability to replace. And that was a characteristic of the tissue we talked about when we did the tissue. So really high mitotic activity. So these guys divide. Where do all the progeny of these stem cells go? Upward. And then go up, 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 up. And the idea is that we'll swap them all and get rid of them. So, when you take a shower or when you just do like this on your clothing, cells are just falling off like crazy. And that takes the bacteria and any other stuff that might have landed on there, old oil that's laying on the top of your skin, all that kind of stuff just falls away with it. That's not an excuse not to bathe, though. Just for the edification of the people. This guy right here is called a keratinocyte. So that's this layer up here. So what happens is, here's our germinal cells right here. And as they divide, then the progeny, when they come up here, they're going to be called keratinocytes. Now, the reason they're called that is because as they move up to the top of the skin, they're going to go through a process called keratinization. And what will happen is we will gradually see the components that make up the normal cell disappear, and we'll go through a chemical process or a biochemical pathway in which we're going to end up making a bunch of molecules, protein molecules, called keratin, right? Keratinization of keratinocytes. And that keratin is a waterproofing kind of tough, hard material, and it will fill up the cells of the stratum corneum. So we'll basically have a cell membrane at that point, and just a big gigantic bag of keratin. And so that's what creates part of that protective barrier, hard to get through those layers and layers and layers of those cells. So they're called keratinocytes because as they move up, they produce this because they produce keratin, and that's how they got their name. Are we okay? Makes sense?
I do like that.
But I hate that. First thing I feel like is I'm in a cave, and second, I can feel my energy level. But you can see it better, I know. The next guy we have here, this is a melanocyte, and a melanocyte produces a molecule, a pigment molecule called melanin. And melanin is a brown-colored molecule or pigment. And so where you see at this top picture, they're showing you the melanin pigment actually around the site of cells, and we can see the melanocytes that are labeled there. And so what they've done is they've kind of drawn a picture here to show you what's going on that you can't see with a light microscope there. And so what happens is, here's our melanocyte, and as the basal cells are producing keratinocytes, these guys have extensions that go out, so like pseudopods, and they're going to go between the cells. And because a membrane is a membrane is a membrane, right? They're all phospholipid bilayers, and they will flow into each other and with each other. What happens is these guys will pinch off little pieces and actually create these little chambers that are full of melanin. So these guys are called melanosomes. You see one being created here, you see one here, one being created here, there's one over here. And what will happen is the melanosomes will migrate to the nuclei of our keratinocytes, and when they get to the nucleus of the keratinocyte, then the melanin is released and surrounds the nucleus of the cell. Now the idea is that if we have this brown pigment and it's all around the nucleus, as ultraviolet light comes through the skin and gets close to the nucleus, these pigments will absorb that ultraviolet light and not allow it to get to the DNA inside the nucleus. If it gets to the DNA, ultraviolet light is pretty high energy, and it will damage the DNA, or it can damage the DNA. And so when we take a look at a lifetime exposure to ultraviolet light, then what happens is we see cells that eventually have enough damage that they can't do their jobs anymore, or we start to see the cells, so because of the ultraviolet light damage, they not only, let me rephrase that. When I say they can't do their job, there's a variety of jobs. So if I'm a basal cell, my job is to divide. I might not divide properly, or I might not divide at all. And then if I'm a keratinocyte, my job is to produce keratin. I might not do that very well, but also the ultraviolet light, if it's deeper into the skin, can damage the layers underneath the epidermis. So the more ultraviolet light we're exposed to, the less productive our dermis and our epidermis is also. And so what happens to people who get a lot of ultraviolet light and don't use sunscreen is when they get really older, they get really wrinklier. They get more wrinkles. Because all of those cells that are supposed to be producing collagen and elastic fibers and everything and keeping your skin all nice and taut and all of that, they don't work well anymore because they've been damaged and your skin looks like an old wrinkly baked potato by the time you're 50 years old. Okay. Does that make sense? Okay. So the more ultraviolet light you're exposed to, the more melanin you produce. So the more exposure you have, the darker you get. And the amount of melanin that you can produce is sort of genetically predetermined. So some of you are really, really super fair skinned, and you just go outside and you're out there for 3 hours and you get a sunburn and you never get brown, and the next day you get a sunburn again. And then some of you are just actually go out for just a little bit, boom, tons of melanin. And then some folks are just naturally have a lot of melanin, so they're already dark before they even touch the sun. It just really depends on genetics. And we'll talk about pigments and stuff a little bit later, but we'll not talk about cells at that point. Questions so far? Okay. So this guy right here, he's called a Merkel cell, and this is also in your notes. The other name for this guy is Merkel-Ranvier cells. They were named after Sigmund Merkel. So he had to be a scientist, because you don't play hockey with a name like that. So he was a nerd from the get-go. So when you take a look at Merkel cells, probably their big role is that they release chemicals that can cause other sensory cells to be more sensitive. The other thing that Merkel cells may do is there's some evidence that when you touch things, they may be able to determine spatial structures or spatial relationships in really, really fine touch. And so that seems to be their two big roles, that really spatial thing with fine touch and releasing chemicals that cause other receptor cells to be more sensitive and more likely to pick up what they're touching and what's stimulating those guys. And so these guys are actually going to be found in the epidermis. Most guys that are going to be involved in any kind of reception or perception will be found below the epidermis. So he's kind of a special guy in that he's this sort of this receptor guy that's actually going to be part of the epidermis, which is very, very unusual. When we take a look at the two kinds of skin, okay, so we've got all the layers in the stratification. Okay, good. So when we take a look then at kind of how this is going to be put together, so here's our strata-basal, and who are the guys that are doing the division?
Basal cells.
Basal cells are germinal cells. Who's moving upward? What are they called? Keratinocytes. What are they going to produce on the way? Keratin. Good. At the top, the cells are alive or dead?
Dead.
Totally dead. And then the guys that are kind of unique and that they're there for some perception of touch would be Merkel cells. Good. All right. When we take a look at how our epidermis and our dermis integrate with each other, what we find is we have this kind of wavy undulation kind of pattern that goes on. And so the guys that you see right here, this is in the dermis, these are called dermal papillae. And papilla just kind of means a bump, and we're going to see that word a lot. And then these guys right here are called epidermal ridges. And these guys fit together, they're interconnected or interlocked with each other, and that's what helps to keep these two guys together. These guys eventually will demonstrate their structure and their pattern on the surface of the skin, like with your fingerprints and your footprints and so taking a look at the layers that we have, that over on the left side you have a stratum basal, and we're going to talk about the stratum spinosum in just a minute, and then we go stratum granulosum, stratum lucidum, and stratum corneum. So this is obviously what kind of skin? Thick, 5 layers. So if we look over here, which layer is missing? Stratum lucidum. Correct. So a thin skin fly still sees the same arrangement of the epidermal ridges and neural papillae, but as we move upward, what we notice is the stratum is actually missing. And then once again, we notice this stratum corneum is not quite as thick as that stratum corneum. There's typically not nearly as much friction, and so it doesn't have to be quite as thick. All right, so the next layer then, stratum spinosum. Oh, here's your epidermal ridges and what we see on the skin because of that. And so our fingerprints, they represent that pattern that you have. Fingerprints are unique or not unique? Unique. Okay, just like your DNA. So anything that comes out with twins as they go through their life, their fingerprints change and they're not exactly the same. So then in the next, where you have the stratum spinosum, we'll take a look at your notes first. So 8 to 10 cells thick of polygonal cells held together by desmosomes. The spinosum means prickly, and the cells appear like miniature pincushions. So here's the deal. What happened, like from this from I can figure out, let's go back for a sec. I'm assuming most of you think that those don't look like pincushions. No. So what happened is early on, when you're trying to take a look at the structure of all of this, the scientists for a long time didn't understand that you could use dyes. And so then when they found dyes and stains that would make these cells show up, they got excited. Well, it turns out that when you take a look at this particular layer, the cells aren't nice little cubes like they are down here anyway. When they get up here, they get multiple sides on them, which makes them a little bit pointed like a stop sign. But it looks, from the reading, it looks like what was happening is the stains that they were using were dehydrating the cells somewhat. And so the cells were sort of shrinking, and then the little edges were sticking out. So they looked like little pincushions, so they called it spinosum. And nowadays, when we look at it, it doesn't look like a pincushion anymore. But we could sort of pretend, because we can see like with this guy, there's a point here, and he goes up as a point here, and kind of goes like this, and he has all these edges, and you can call each one of those a little point. You can make spinosum, spiny, and in your imagination, I don't know. Does that make sense? And if that doesn't work, just remember it's spinosum and get over it, I guess. Now, in this layer, you've got a guy called the Langerhans cells. That's what all those little arrows are pointing to. This is one of those where they've changed the name, and the new name absolutely sucks. Although it's a great name. So here's not you're not going to suck to be a great name at the same time. It really is a bad name because until you understand what these cells do and all their roles, the name doesn't make any sense at all. Once you understand what the cells do, then they make sense. So they named it to describe it really well, but for the average person, this descriptive name is just something they're going to hold out with that name. So here's the deal. If you, on a test, you call these the Langerhans cells, it's okay, because Langerhans is easier to remember and it's easier to spell. And if you hear this other name sometime in your life, in your career, at least remember what it is and what it goes with. So, the other name is non-pigmented granular dendrocytes. So here's what this means. When it says non-pigmented, it doesn't have any pigment molecules in it, so it doesn't pick up melanin and those kinds of things. Okay? It says granular because when you stain it, the stain grabs onto some of the proteins that are there and gives these guys kind of a grainy-looking appearance, much like the granulosum that we'll see here in a little bit in our skin. And then the word dendrocyte is a specific kind of cell that we have in our body that's an immune cell. It's a defense cell. So the job of the Langerhans cells is to cruise through the epidermis, or sometimes they're fixed, so sit there and wait. And what they want to do is they want to come in contact with the pathogen. Now, once they come in contact with the pathogen, then what they can do is they can take and engulf that pathogen, or if they come in contact with like a cancer cell, you get one of your cells that's starting to get a little wacko, they'll engulf those guys and try to kill them and eat them up. So if they came in contact with a pathogen, let's say a virus got inside of you, they would engulf the virus, and they would move some particles from from that virus up onto the membrane of the cell. And at this point, what they want to do is they want to go find a special cell in your body called a helper T cell, and they want to show that helper T cell those virus particles. Because once they do that, that helper T cell will agree with your Langerhans cell that that's a bad guy, and it will send out messages that will gear up your entire immune system to now attack that particular pathogen that you have. Now, the guys that can engulf something, move out of the tissue where they belong, move into your lymphatic system and possibly your circulatory system, and cruise through your body looking for that helper T cell, those are called dendritic cells. So that's where the word dendrocyte comes from, because these guys are dendritic cells. They can leave your skin and go out to other parts of your body to find that helper T cell and say, hey, take a look at this guy, I think he's bad, and helper T cell goes, ooh, he's really bad, and let's tell him what's in the system. Did I just lose you, or are you okay?
It's kind of like a flight messenger looking for the person that needs to serve the papers.
Or he's more, have you ever read Are You My Mother by Dr. Seuss? There you go. He is the little bird that's walking around going, Are you my mother? And so the difference is he's got a little virus part going to all the helper T cells and going, Are you my partner? and you tell me that this is a bad guy. Do you agree with this? We're getting infected. Let's get with the program. Make sense? All right.
What produces
The Langerhans cell?
them?
So actually, these guys are can go through mitosis themselves. They're also a product of your immune system. There's just some cell lines that along those lines. Are we okay? All right. So once again, he's also the guy that protects you against cancer and stuff. So the next layer you have is the stratum granulosum. And what we notice about this layer, not particularly thick, but notice how dark it is. And on this particular slide, they're trying to get the whole epidermis in the view, so they didn't go down with their higher magnification. But if you look at these cells in the higher magnification, they stain really, really dark because of the process of keratinization. And so the molecule that's there, and because they've gotten kind of dehydrated at this point, that really, if you put stain on it, it grabs the stain and really holds onto it, and you have all of these dots of stain inside the cell. It's grainy, so they call it granulosum. That's simply how it got its name. So it says 3 to 5 flattened cells, thick. Cells have a granular appearance to the production of keratohyalin. So keratohyalin then is going to be an intermediate molecule in our process to make keratin. And it says keratohyalin promotes dehydration of the cell and linking of keratin fibers that are surrounded by keratohyalin granules. And those are the guys that grab onto the stain. Does that make sense so far? All right. So at this point, our cell is really on his way out. We've got a degradation of the organelles of the cells. We have a lot of keratohyalin that is going to eventually be keratin, and so our cells are kind of gone at this point. Does that make sense? Okay. On the next layer, it's the stratum lucidum, and so it's this clear layer next to it. And once again, 3 to 5. Oh, I should stop. Numbers are only important if I tell you they are important. Are we okay? So the stratum basal is like 1 cell layer thick. We're all just going to agree it's 1 cell layer thick, and that's kind of an important number. The stratum corneum is going to be really thick, like 30, 40, 50 cell layers thick. And as long as you know it's the thickest one and it's really thick, we're okay. And you recognize if I say what layer is 30 to 40 cell layers thick and give you some other descriptions, you can fit a stratum corneum. But you do not have to memorize these numbers. I'm not going to ask these numbers on the test. I don't know why, but students, maybe because you take math, or maybe your history teacher did this to you, but students think if there's a number, they should memorize that number. We have important numbers and we have who cares numbers, and so I'll tell you if the number is important. And so these are important, except you just can't understand sort of the proportion of the numbers. Okay? So it says clear appearance because the nuclei and organelles have disintegrated. So are these guys alive? No. And in fact, everything's broken down so much that it's really hard to get steam to grab onto what's left. It turns out that the molecule that we're creating now is called elegin, and that's going to eventually become keratin. That doesn't hold steam very well. So what we end up with is this layer that just really doesn't have much steam attached to it, except sort of on the cell membrane and a little bit inside the cell. And light, because there's no organelles and things, just small proteins, light goes right through the cell, and we don't see any image because the light's not moving and bouncing off of things. And so our retina can't pick up an image because there's nothing to catch for the light or stop the light. Does that make sense? Right? So the next layer is the stratum corneum. The cells are now full of keratin. They're surrounded by the plasma membrane. They're dead. They're held together by desmosomes. And when they fall off in sheaths. And if you're not convinced they fall off in sheaths, most of you have had a sunburn, and you've got the little water blisters, and you go to pick at a piece of skin, you grab it and go like this, and a big, huge sheet of cells comes off, right? So, stratum corneum, so if you're pulling off. Make sense? Okay. Okay, the next thing that we need to talk about is vitamin D production. So you probably want to kind of write this down. It's not in your textbook. So what happens is the sunlight, ultraviolet, comes through the skin, and we have this molecule called 7-dehydrocholesterol. So let's stop for just a sec. Cholesterol is the molecule that, when it says cholesterol, that you probably think about in terms of I don't want cholesterol in my body. Reality is your cell membranes are all made of cholesterol. A significant number of the steroid hormones that we have to start out as cholesterol molecules. Cholesterol is a good guy. So this happens to be a form of cholesterol that's important to us, so we can't make a vitamin D. So the problem with cholesterol is you don't want too much cholesterol. Your liver is purposefully going to make cholesterol. It's that when our liver goes nuts that it causes us problems, or on top of what our liver is producing, which meets our body's needs, we eat tons more cholesterol. So we're going to start out with 7-dehydrocholesterol, and all the 7 means is that when we have organic molecules, so those are ones that are carbons, hydrogens, and oxygens, we have functional groups coming off of those. And we want to designate which carbon the functional group is on. And so it's saying on the 7th carbon, we have a hydroxyl group that's on top of us. That's all that fancy stuff actually means. So the ultraviolet light then is going to cause this to be converted with some enzymes to a molecule called cholecalciferol. And so this is what our fancy name for vitamin D3. What will happen is vitamin D3 then will go to the liver, and the liver will modify that molecule, and it'll make a molecule called 25-hydroxyvitamin D3. Once again, it's just saying on a particular carbon we have a functional group that's there. So it's vitamin D3 with a special attachment. Let me stop for just a second. We're going to use functional groups a lot over the next 3 terms. Is there anybody who... Let me ask this. How many people feel real comfortable with the term functional group? Okay, that was a better way of doing that. So it didn't embarrass anybody. All right, back into 11 real quick. They talked about functional groups, and they talked about an alcohol group, which was in OH, or a carboxylic acid group, a COOH, or an amino group, an N with 2 nitrogen with 2 hydrogens. And you attach those to different molecules, and they cause their physical and chemical properties to change. And so there was a whole list of them in your 211 textbook. So when we use the term functional group, what we're saying is one of those molecules has been attached to another molecule. And so sometimes you might recognize them, sometimes not, and sometimes they're really important to know them, and sometimes it's not. In this case, we don't care that the functional group is a hydroxyl group. We just want to recognize that we just changed the structure of this guy to this guy. Are we okay? All right, so we don't care about the chemical composition. The name's good, and that's fine. So this guy then is going to go to our kidneys. And what will happen at the kidneys is the kidneys will convert this form of D3 to 1,2,5-dihydroxyvitamin D3. We've added a second functional group in the first carbon position. And so now what we've created is a molecule that we generally never use this term, we use this term. So we created the molecule calcitriol. And calcitriol is really important in helping us to maintain our appropriate calcium levels. And so vitamin D3 then becomes real important in maintaining an appropriate amount of calcium so that you have healthy bones. Does that make sense? All right. So if you're going to make vitamin D3, you got to get what? Sunlight. Now, having said that, for most people, the amount of time you spend just walking from your house to your car and your car into work and back and forth is enough sunlight to get enough ultraviolet light to make enough vitamin D3. Are we okay? So you do not have to go out and sunbathe to get vitamin D3. Your neighbors do not want to look over your fence seeing you standing out there naked, like vitamin D3. That kind of thing. At certain times of the year in Grants Pass, like now, we don't have a very high sun that's coming in at a weird angle, and the amount of ultraviolet light we get is a little bit less than really what we need. It's only for a few weeks that happens. But fear not, because we can get vitamin D from our diet, and they fortify all kinds of foods with vitamin D. So once again, you do not have to run to the tanning booth and you get the ultraviolet light there, because that's going to cause you to get wrinkly and have skin cancer. So stay away from that place. So dietary intake, if you eat meat or take supplements or if you drink milk, those all have vitamin D in them. Be careful with supplements. Vitamin D is a fat-soluble vitamin, and fat-soluble vitamins can become toxic if you take too much of them. They store it in your adipose tissue, and then you can get what's called vitamin toxicity. And so you don't want that to happen. Does that make sense? Okay. All right, now, what happens if I don't get vitamin D? So vitamin D was to help us keep what mineral level up? Calcium. And calcium for strong bones. So here's a person who didn't get enough calcium. The mineral part, right, the calcium part of bones is what gives us its firmness and rigidity. And so now this guy's got too much collagen, not enough mineral salt, and the pressure from his body or her body is causing the legs to go out. And so this is called rickets, and there's evidence of rickets here, and lack of vitamin D in their diet. Okay? And you don't see rickets too much in the United States. All right, so that's kind of your epidermis so far, and vitamin D and stuff. Are we okay so far? All right, last part of this is a molecule called epidermal growth factor. So, as the name implies, epidermal growth factor probably does what? Promotes growth, and probably in the epidermis. Good. And so if you take a look, it says peptide hormone. We'll talk about hormones next term. You'll figure out the difference between the different classes. So it's just a peptide hormone produced by the salivary glands and glands through the duodenum. Everybody I'm sure knows the salivary gland. The duodenum is the first part of your large or small intestine. It's connected to your stomach. It's about 12 inches long, and we also produce this molecule there. So what it does is promotes division of the germinal cells of the stratum germinativum and the stratum spinosum, and it says accelerates the production of keratin, and so stimulates epidermal development and repair, and then stimulates synthetic activity and secretion of epithelial glands. So far, everything we've talked about is helping with the growth and repair and maintenance of what structure? The epidermis. That's called epidermal growth factor. The other thing we use it for, it says used to promote growth in tissue culture. So clinically or with research-wise, what happens is, and so when you talk to people and they say, well, I don't know why we just can't grow that, the first thing you can tell them is it's really hard to grow tissues in the lab. the cells don't want to grow normally or in the lab. So in the lab setting, we're trying to do artificial tissues, especially like skin. When the cells get placed on a medium where they grow across the medium, they get to the edge of the container, the cells start to get squashed a little bit, and a normal cell, that's his signal to stop growing. And so that's why your body just doesn't keep getting bigger and bigger and bigger, and your organs don't grow forever. Is when they get to just the right size, they're getting squashed a little, and then they stop going through mitosis. Now, if 100 of them die, that frees up a little room, and so as soon as some guys are going to divide, so we create them out of the right number, and we get squashed again, and then we stop. And so with epidermal growth factor, what you can do is you can put this on all kinds of different tissues, and you can get those cells to grow again. And so then they'll grow on layers, which is what you need if you're going to make different kinds of tissues. Does that make sense? The lack of control of growth is exactly what the problem is with cancer cells. They just grow. And so we'll talk about cancer a lot of times, actually, throughout the 3 chapters. That's one of the big differences between cancer cells and regular cells. So the next layer that you have is the dermis, and it's broken up into 2 components, the papillary layer and the reticular layer, or the stratum papillorosum, stratum reticulosum is the other terms that we use. So what we do with this guy is basically say the top 1/5 is the papillary layer. There's no real physical sort of obvious demarcation between one and the other, but what we've decided is, structurally about the top 5, the top 20% is called the papillary layer, and everything on down is going to be the reticular layer. And then what happens is, for you guys in lab, when you're trying to label these guys, if you're looking at a picture, where does the reticular layer of the dermis end, and where does the hypodermis begin? So the easiest way to do this, and it's not perfect, but what we've kind of all agreed on, who teach this class, is once you see adipose tissue, you are in the hypodermis or the subcutaneous layer. So that's going to be our big kind of clue of where we're at. And so once you see that, just say, okay, hypodermis, I'm safe. And that's going to be our physical sort of evidence of where we're actually at. So what we notice about the dermis is there's a lot of accessory structures that are here, there's a lot of sensory structures that are here, and there's a lot of vascularization that's going to be present here. So very, very different than the epidermis. What happens is for the epidermis, one of the reasons he can't support all of this is because the blood supply ends right there. And so all of the nutrients, all of the oxygen have to get to the epidermis through diffusion and osmosis, and that limits how thick it can be, and it limits what it can support. Are we okay? All right, a couple other house cleaning things we need to do that usually students get a little confused with. In the book, they'll talk about where things are derived from and where they are located at. And sometimes that messes things up for folks. And sometimes the authors mess things up. So if we follow this purple, this is all epidermis. What do we notice happens here? Yeah, it goes deep into the dermis, doesn't it? And we're going to actually create this hair follicle and the hair from epidermal tissue. And so, truly, that is an epidermal derivative. Are you with me? Some of you aren't shaking your heads, so where did I lose you?
So it's part of the epidermis. It goes around the hair follicle.
Yeah, what happens is all of these cells are epidermal cells, and they're going all the way down in here. So these cells are actually creating these structures.
So it would be squamous around those then. So
Well, no, these are all going to be like cuboidal cells and things.
Okay.
So you would say the hair follicles derived from the epidermis?
Yeah, technically it's derived from the epidermis. Now what happens is, this is where the authors kind of get really weird. Sometimes they'll ask you, where's this located? And some authors will say it's located in, so if you're reading other books, they'll say it's located in the epidermis. But no offense to these guys, any fool who looks at that can see that it's plainly in the dermis. Are we okay? So what we do in our class, so if you're reading other books, so that they don't start confusing you, we'll read that it's derived from the epidermis. It truly is. So in terms of location, this guy and this guy and this guy, the little sebaceous gland, they're in the dermis. It's just the epidermis comes down into the dermis.
Her hair derived from the epidermis is located in the dermis.
Yes.
Okay. As well as the sebaceous gland.
Yes. Perfect. Well, does that make sense? The follicle surrounds the hair. There's an area called the matrix. There's part of the follicle that makes the hair. Don't split hairs. That's pretty good. I am using myself. So here's your sign. Yeah, there's your sign. So if you look at the stratum papillosum, so the upper layer, 1/5, we already did that. Marked by projections called dermal papilla, we talked about that. And this, we might as well talk about all these things really good. In this layer, what you'll see is this guy right here. The new name for that guy is a tactile corpuscle. The old name for this guy is a Meissner's corpuscle. I think your lab book is still using Meissner's corpuscle for this guy, but this is the new name. Tactile corpuscles or Meissner's corpuscles are for light touch. So if somebody were to take and lightly run a feather, across your skin, or if you walk outside and there's just a hint of a breeze and you can feel it on your cheek, that's the tactile corpuscle that's doing that. Does that make sense? Okay. The other guy we see that's down into the reticular layer, so these guys are very, very prominent. There's a lot of these guys. The other one we see is this guy right here. So his new name is called lamellated corpuscle. And so he's called lamellated. Comes from the word lamella, which is layers, like plywood. And so when you look at this guy, it's really what it always kind of reminds students of most of the time is if you took an onion and you sliced it in half and looked at the inside, you have those layers of onion. That's really what this looks like. So that's how I got its name, lamellated. It's got these layers that are here. His other name is a Pacinian corpuscle. Pacinian corpuscle. That's the older name, Pacinian. And I think I got those in your notes. And I think, once again, your lab book is using Pacinian corpuscle. So he's deeper and he's touched. So he's for deep pressure. Yeah, deep touch. So if somebody comes up and lightly does this on your skin, he's doing that. Meissner or tactile. But if somebody comes up and squeezes your arm, that is lamellated or Pacinian. They just have two different roles. All right. Okay. In the reticulosum, what kind of tissue and fibers are going to dominate? In the reticulosum, what's the dominant fibers and tissue that we see?
Elastic
Connected and dense, regular connected.
So loose connected and dense irregular connective tissue. We talked about that already. They should be irregular. Is that what regular?
Yeah.
No, it's irregular. That's not good.
No, it's not.
And we thought, remember when we did the other day? Somebody over here was the volunteer, and we went like that on their arm, and it goes all different directions because of the irregular connective tissue. Are we all right? Okay. So if this guy's going to be strong, keep your skin from pulling apart, and that irregular connective tissue fiber would probably be, what was the dominant fiber?
Collagen.
Collagen, thank you. That's our strong fiber. There's a lot of collagen in the process. Are we all right? And what happens when collagen is exposed repeatedly to ultraviolet light?
Breaks down.
Breaks down. So So the firm stuff is holding your skin in place and making your skin firmer because you don't produce as much collagen when you get older, and ultraviolet light damages it, means your skin is going to sag a little bit, and the more ultraviolet light, the more nastier it's going to sag and look. We're building up to the concept that there's no such thing as a healthy tan. That's what we're building up to. What?
Is that one of your other soapboxes?
Not particularly. I'm amazed at the number of people you meet that are complaining about their wrinkles as they, talk about their next appointment at a tanning booth. It's just like, here's your sign. Right? So I'm just telling you, the big question everybody asks is, how can I live longer? Right? And so the plan, because it's A and P. And how do I make these diseases and things, how do I not get them? How does my body not decay and get worse? And the reality is that it is. And you're going to die. And that's just the way it is. But really your goal then is to just delay that as long as possible and be the healthiest, best looking corpse possible. and so I'm just giving you some ways to do that. Okay? So one of the things you don't want to do is play around with ultraviolet light too much. And that's why especially for the women nowadays, you go by with makeup and you look at it, there's always something that says like SP5 on it. Sometimes they're even more than that. They're just building it right in. That'll save you a lot of dollars later than these Botox injections. Are we okay? All right. So, taking a look at some of these other guys that we have here, one of the things that we'll notice here is we've got these guys, which are erector pili. And so, when we say erector pili, this is spelled with an A. And so, erector pili, they'll spell it with an E. These guys are attached to our hair follicles. When they're stimulated, they contract, and it causes our hair follicles to be pulled up, and our hair stands up. And so that's a defense mechanism. When you take a look at other animals, when they get frightened or they want to be threatening, these little muscles contract inside of their skin, and their hair stands really big up. So your cat is really angry with you, or there's a dog in the yard, and the cat goes, right, and does like this, and brings its back up, and all the hairs go out like this. The other thing it does is then it makes the hair real sensitive. So if something brushes the hair, it will pull on these guys, and the nerve endings here will kind of pick that up. And so then they can sense around them really well. So when you get frightened or nervous and stuff, sometimes you get goosebumps or goose flesh. But it really doesn't make you look threatening at all, and it really doesn't do much for your sense, figuring out what's going on in your environment. It's just a holdover that all mammals sort of have, and yours just doesn't work that well. But sometimes, it still occurs. The other thing that we'll notice with this guy is that we're going to have some regions where we have vascularization that occurs. So in our papillary region, we have the papillary plexus right here, and then in the hypodermis, we have what we call the cutaneous plexus. So we did our basic pathway of heart, arteries, capillaries, veins, heart that first week, correct? So what we're doing is we're following that pathway in that when we get down here, when you see the word plexus, this word plexus means that you just have kind of an interweaving or a network that occurs. And we're going to use the word plexus a lot. So what we're saying is you have a lot of these vessels kind of interwoven here, and they're branching off, kind of creating this weave that's going to move up this direction. And then what we're seeing here is a group of capillaries. So they're capillary beds is what they're called. And we're calling this a plexus because there's a whole group of them that are kind of intertwining this presence there. And so what happens is the blood comes into the hypodermis. A lot of blood vessels are branching and going in different directions. Some of the blood vessels move up this way, so they move up, they branch, the dermis, and then these guys come up here, and they form our capillary networks, and diffusion and osmosis brings the material up here, and diffusion and osmosis brings the material down here. So all of these cells need to be fed, oxygen's leaving, nutrients are leaving like glucose, and then these guys are busy going through metabolic activity, so they produce CO2 that goes this way, and they produce organic waste products from those biochemical pathways, and they go this way into the blood. So you have this exchange that's going on. Does that make sense? Are we okay? All right. So some of the other guys we see are sebaceous glands, and we're going to talk about some of these guys in more detail by themselves. We're just doing an overview now. Sebaceous glands or oil glands. When we take a look at sebaceous glands, typically we see them associated with hair follicles, but they do not have to be. But their big job is to produce oil. It keeps the hair supple and soft, and it keeps your skin supple and soft. So that's really one of its big roles. And then when we also take a look here, we'll see sudoriferous glands or sweat glands. That's these guys that are right here. One of the big groups of these guys, the ones that you think about as being sweat glands that produce the perspiration that cools you off, these are called American sweat glands. And so they release their secretions through exocytosis, through the cell membrane. So they're actually called American sweat glands because of the way they release their secretions. And we have another kind of sweat gland we'll talk about in just a little while.
Sebaceous glands be holocrine?
In the sebaceous glands, yeah, those are sebaceous or holocrine glands. So the whole cell dispersed. Yeah, exactly. Are we all right so far? All right. A couple other endings, nerves that we have up here. This is a free nerve ending right here. It's just the nerve that's coming up. And remember when we did our axon and our dendrites, right? Those little extensions? Okay. So what happens is those little extensions come off the cell. In this case, they're going to be dendrites. And they're just sitting right here. And if it's a free nerve ending, it's typically going to be for pain. That's the primary role of free nerve endings. The other guys that you have up here, and I don't see them actually listed on here, you're going to have some guys that are called thermoreceptors. And so what happened when they were taking a look at how they were naming all of these receptors and stuff, there was a little bit of overlap of naming, and then since then they've corrected it. So if you take a look in your notes on Roman numeral 3, and it'll be numbered kind of at the top of the page. It has organs of Ruffini listed there, and then behind it says thermoreceptors. Everybody see that? Okay. So this is hypodermis right here. If you move up to here, it says organs of Ruffini thermoreceptors. Okay? We're going to make a change, because now they've cleared up their problem. So we used to have, well, once again, we have all these guys that did all this research. We had this guy named Ruffini, and this guy was a wild man. And so he was studying all kinds of nervous system stuff and naming stuff like crazy, and so they named a lot of stuff after him. And so we had organs of Ruffini, and we had Ruffini corpuscles. And one guy was supposed to be for temperature, and one guy was supposed to be for pressure. And what happened is everybody kept interchanging the names. And so you'd be talking about the pressure guy and using the name for the temperature guy, because the temperature guy was also being used for the same name as the pressure guy. And so somebody finally decided we have to stop the madness. And so what they've done, it's a Ruffini corpuscle. So you can cross out organ of Ruffini. Just write, so you see Ruffini, underneath write corpuscle. Cross out thermoreceptor. And then write down pressure and distortion. So if we have a Ruffini corpuscle in your skin or anywhere else, it's sensitive to pressure and distortion. Are we okay? Alright. So you get to be my guinea pig right now. Okay. So I'm going to touch your hand. Do you see your skin indenting? So tell me what two receptors, or you guys can tell me, what two receptors respond to that? I got a big dent in your skin. Or laminated and Ruffini corpuscles. Pressure. Are we okay? Distortion. Are you with me? Okay. If you have a receptor that responds to temperature, what do we call it? Thermoreceptor. That's our name. Thermoreceptor. We just made that really simple. Thermotemperature receptor.
Thermoreceptors are thermoreceptors.
Okay. Thermoreceptors are thermoreceptors. Changes in temperature. And that's really, when I say changes in temperature, that's what they do. They really don't send you signals unless the temperature actually changes. So if you move from outside cold and you move into the building where it's warm, they immediately send you a sensation and you go, ooh, it's warm in here. And then when you move from the building outside tonight, half you're going to go, ooh, it's really cold outside. Because the thermoreceptor told you it's really cold. There's a big temperature drop that occurred here. And that's what they tell you is change in temperature. Make sense? So like in the summertime, if you ever get the summertime again, when you're in a restaurant in the middle of July, and all of a sudden the air conditioner kicks in, and you're sitting by where it blows out, and you go, ooh, there's a drought thing where the air conditioner is still on. Thermoreceptor just shows you temperature changes, blowing cool air on. Okay, so we did lines of cleavage. These are basically a pattern that we have created that reflects the way that the fibers are primarily running in your skin. And so this is collagen. And what happens with this collagen, when you have these dominant sort of directions that things will run, if we cut across that line, the pressure is going this direction and this direction. So it's pulling, right? And so if I cut across that line, and then I sew this back together again, what are those fibers going to be doing to that incision? Pulling it apart. And so if we can cut along the lines of cleavage, then what would happen? Yes, instead of my fibers pulling this way, they're all running this way. So I'm just cutting between them, spreading them apart. I'm not cutting across them, right? Just spread them apart, take out what I need, put them back together, and there's no pressure to pull that incision apart because all the pressure is going along the edge of the incision. Make sense? So, let's take a look. This is this guy's, what's this part called? And the break-in. Good. And it makes sense that the collagen in the skin is going this way because which way is the muscles moving? Primarily this way, aren't they? All right. So, a few weeks ago, I went in and they decided they needed to remove something from my arm. So I don't know if you can see that. So you see that scar right there? Okay. So now what happened was, where's the lines of cleavage going? This direction, right. And so they cut which direction? Right across. And so when they did that, they said come back in about 12 days. And I said, sometimes when I get incisions, they don't quite heal that fast, so let's shoot for 14. And the doctor said, okay, 14's good. Turned out we couldn't make the appointment for 14, so I went 17 days to take the stitches out. That turned out to be really, really wonderful because it healed for a whole lot with the stitches in. The inside stitches were still there, and then they told me for another week, don't do anything too strenuous. So this was my first strenuous week. But because we took care of it, and it went a little longer with the stitches holding it all together, no separation. Because the whole time, the lines of cleavage are trying to pull it this direction. They couldn't cut it this way because of what they were trying to remove, so they had to go that way. Does that make sense? So if you can cut along that, it's usually less scarring, it's less pulling. So it may not make that big a difference on your forearm, but if you're a plastic surgeon, big difference. You really want to do that if possible because you're getting a lot finer scar that way. Cool. So this is just for that dermal blood supply. Kind of showing you a little bit better picture of this. So notice we have our cutaneous plexus here. We have deeper blood vessels here. They lead up to here. There's our network. These guys lead up to here, and there's our subpapillary plexus network that's up here. Everybody visually can see how that works. We haven't done blood vessels, so I'm just trying to get you a picture of how it kind of looks. Same idea. As you can see, the blood vessels that are here. With the hair, we're going to talk about these guys in a little bit. Notice there's the blood supply to the hair. there's a little area right here in here called the matrix. This is called the dermal papilla right here. And this matrix is where all the cell division is going on for that hair to grow. So we need to have the blood supply really close to that because there's a big demand for oxygen and nutrients from those structures in that matrix. And notice, here's our sweat gland. There's a capillary bed going to the sweat gland to make sure it's got plenty of oxygen and nutrients taking away all of the waste products. Capillary network that's here also. So who's doing the work of the circulatory system? Capillaries. The tiniest blood vessels you have do all of the work. The other guys are just transporters. The heart, just the pump. All the exchange, that's the capillaries. Cool. All right. So hypodermis, primarily going to be adipose tissue and areolar tissue. Good. And so why would those be two good choices here? So it helps hold things together. It's a whole bunch we just said insulation, right? Padding, cool. And the areolar tissue, when you look at its ground substance, it was sort of, kind of syrupy. And was there a lot of it? Yeah, so the fibers themselves were really spread out. And we said that was a really excellent source of it's a great place to store stuff like electrolytes and those kinds of things. And so what happens is that all of the areolar tissue here, and we can store electrolytes. So electrolytes are all the ions that your body needs: potassium, sodium, magnesium, zinc, all of that, chloride ions. So excellent source of storing all those guys if they're needed for the cutaneous layer. So, that makes sense? So this is taking a look at the wrinkles on your skin. These are kind of the two kinds of wrinkles that we have. So when you take a look, this is a congenital pattern. So what does that mean? You're born with it. And so when you take a look at a newborn, where are those located at? Fingertips anywhere else? Toes anywhere else? Palm of your hand and sole of your foot. I don't know if they still do it. It used to be when we were having kids, they took that whole foot and slapped it on a pad and stuck it on there right after the baby was born. So no mix-ups. And if you're really just want to be tough, say, I want you to match the footprint before I take them home. Then you have to go dig out and do the whole thing. But this is just trying to show you. And did you guys do this when you had lab?
It was optional.
Oh, it was optional. Did you do it? Oh, it should have made it optional. It's cool to look at your fingerprints and then try to figure out what all your different patterns are. So what happens when we talk about a fingerprint? We've basically taken some basic patterns that we find in all fingerprints. We've labeled those, and then what happens in your fingerprint, you typically have a combination of these guys. And so that unique combination of whorls and loops and simple loops and all of that is what makes your fingerprint your fingerprint. And so it's really kind of neat. It's all genetically predetermined. So those are the ones that you're going to be born with. What are the rest of them? What's in your notes? Take a look. There's a skin pattern. So acquired, what does that mean? You got those for being alive. So that's your reward. So if you're really old and wrinkly, you earned every one of those, and that's your reward for living that long. So it says surface lines begin as they grow older, so flexion creases, flexion lines, and so flexion creases. If you take a look at here in the anticupal region, or here in the carpal region where we bend, right? We've all got these little creases. Now, if you take a look at a baby, and you can't count the chubba little creases. Okay, that baby's supposed to have a little chubba stuff on their ankles, and so that's not one of those creases. Those guys don't have any of this. But when you start doing this, you start making those creases, and those are acquired kinds of patterns. And So the older you get, the more movement you make, the more of those that you end up with. So some of you, after laughing a lot, because when you laugh, you squint it right, and then it makes some lines here. So we're going to call them laugh lines and not crow's feet. And so as you live, you go like this all the time because you're smiling and laughing, you get those little wrinkles that are right here. And some of you are doing like this happening, you've got all these wrinkles across the front. You're doing that for a long time. What's interesting is those are all, no matter what, those are called acquired kinds of lines or patterns. What's interesting is, in some ways, they're genetically determined. So you got your structure from mom and dad, and if you look at mom and dad long enough, and then so if your mom and dad, my dad is famous for this one. Just this brow would come down, two huge wrinkles right here. You look like this after you, it's like, oh man, I'm in trouble. But so when my dad got really old, two huge, deep lines right here. Well, every once in a while, get like this and frown, and I realized, oh. And I started rubbing right here. So what'll happen is, as you get older, you end up sort of seeing some of those wrinkles that your mom and dad have in your face because they set your pattern, and if you make similar faces, you're going to create the same kinds of wrinkles. Are we okay? Does that make sense? Okay. What? Yeah, when they look up and go, how come Dad getting the stinking wrinkle? Alright, so acquired skin patterns. So George Clooney and his forehead. What was really interesting about this picture is, first off, I like her as an actress, but they were talking about how she wasn't afraid to let her laugh lines show. And I look at the picture going, what? But she does have a few right here. So apparently that's a big deal for those little diamonds in Hollywood. This guy's got some cool ones because he has that great smile. This lady, God, who knows? I like this lady because she's smart. See, she earned all of these wrinkles. She's really old, but she wears a hat. So if she didn't wear that hat, she'd have more wrinkles because she gets the ultraviolet light. So she did a nice job. And this is what happens if you've taken a Botox. You either don't have any wrinkles or you make a really great looking corpse. One or the other. You could be either one. Making sense? Are we okay? Right. And now we're talking about pigmentation. So notice, big, huge scale of sort of pigmentation. So what you're going to out here is you have 3 skin pigments. The book is technically wrong. So you have 2 pigments that are actually produced by the skin, and you have 1 that's part of your blood that can cause a change in the color of the skin. So we'll talk about 3 skin pigments, but only 1 of them is produced actually by the skin. So one of those is called carotene, and carotene is sort of a orangish, yellowish sort of pigment. So we have carrots, and carrots are full of carotene. So when we take a look, we all produce carotene. It's kind of important for epithelial maintenance, but when we get to the special senses next term, I always point out that he's very important for the retina. Our color vision is dependent on this pigment, so he's an important guy to produce. But what that does is it gives the skin kind of a yellowish, orangish sort of hue. Everybody produces carotene. Some people produce more carotene than others, and that's why when you go across the globe, you see people with some different hues of skin. So typically, if you go towards Asia, there's more carotene produced in Asian cultures or people that you wouldn't see maybe in Norway and Sweden and those kinds of folks. Are we okay? Okay. The other pigment you see is melanin. So melanin's the brown pigment. So if we go up to Norway and Sweden and all those places, a lot of folks with melanin, right? No, really they don't produce a lot of melanin there. If we go down towards the equator, folks have more melanin. Okay, now, what happens is when we talk about skin color, back in 2011, they talked about this a little bit, and so this is a good spot to kind of fill this in again. They talked about Mendelian genetics, right? Dominant, recessive. And so Mendel was just so lucky he just happened to pick the right plant and the right traits. Because it had one gene from mom and one gene from dad, and one was dominant and one was recessive, and it worked just fine. As you read through that genetics chapter, though, you had polygenic inheritance and multiple alleles and codominance and all that kind of stuff that went on. And it turns out an awful lot of our DNA and our genes work in that other kind of format other than just two alleles for each one, dominant and recessive. When we take a look at skin color, so you see a variety of colors that are here. I really like this one because it really kind of shows it. That's because there are multiple alleles for pigmentation. And so what we have is we have alleles that say produce melanin, alleles that say basically don't produce melanin or produce very little. And your combination of those is what determines your skin color. So if you get a lot of those, well, I think it's about 12 of them, I think, yeah. So if all 12 of yours say produce melanin, your skin's going to be really dark. If 2 of them out of 12 say produce melanin, you're going to be really light. Does that make sense? It's just a matter of the combination that you got. In this case, this is albinism. The people who suffer from this actually have perfectly good genes that say produce melanin. Where their problem is the biochemical pathway to produce melanin. There's multiple steps to make that molecule. And in one of those steps, you need a specific enzyme called tyrosinase. And those people have a genetic issue where their DNA doesn't code for that particular enzyme, so they can't complete the production of melanin. Their body wants to make melanin, but it lacks that one enzyme to do it. And this is what you end up with a person who just really doesn't have any pigment whatsoever. This person has obviously, those genes are almost all of them, or a lot of them are saying produce melanin. And so this lady has freckles, but she has more melanin than this lady who has freckles. Freckles are just where we have aggregations of melanocytes together and they're producing melanin, so you get more melanin in one spot, and so instead of your tan being even, you get little speckles in your tan, because it's little darker spots that are there. And then we have, this spectrum of the colors you can be, and then we have Michael Jackson, who actually had a condition where he stopped producing melanin, and so he got lighter and lighter and lighter as he got older. When we take a look at the idea of melanin, we don't need this for a second. The original thought was melanin was important to prevent skin cancer, and it is. But that's probably now not the reason that we see variations in melanin. That's not the current thought. Relax. We got 3 or 4 more minutes, and sometimes this shows up on the test. Pay attention. So what happens is our current thinking is that this is a function of folic acid, ultraviolet light, and maintaining DNA and mitotic activity. So it turns out that when you're exposed to large amounts of folic or ultraviolet light, and you have vitamin D, that helps to create folic acid. But too much ultraviolet light breaks down folic acid. Folic acid is critical in the formation of DNA. In order for DNA to make copies of itself, you have to have folic acid. So if somebody gets pregnant, what will they tell them to eat? Dark leafy, green leafy vegetables, because they contain a lot of folic acid. Or they'll give them a supplement for folic acid. And so what happens is, if you take a look at places where you have high ultraviolet light, it is a benefit to be dark skinned because you produce just the right amount of vitamin D, therefore folic acid, without the risk of breakdown of folic acid by the vitamin D. If you go up to Norway and you're really dark skinned, then you don't absorb ultraviolet light very well, so you have a dominance of people who are more fair skinned, because what happens up there, fair skinned people would have survived and had children, dark skinned. Now, we're talking about before you had boats, planes, anything where we could go across the globe, hunter-gatherer folks in small groups, And so in Africa or South America, you wanted just the opposite. The folks who were pasty, they weren't producing folic acid, they never carried fetuses to full term, they weren't reproducing, so pretty soon the light genes disappeared, dark genes were left. Norway, dark genes disappeared, light genes were left. Nowadays, diet will help take care of that, so now we can kind of live wherever we want to and go all over the place. Okay, does that make sense? Okay. Two are produced by the skin. Those are the two. The third one that you have is hemoglobin, and that's a result of vasodilation and vasoconstriction. And it works really good when people are fair skinned. It doesn't work so good when you're really dark skinned. So if you get a fair skinned person that gets really frightened, you have vasoconstriction, and they get pale, there's less blood to the skin. If you go out and you exercise, you get flushed and you look red because you have more blood going to the skin, and the hemoglobin pigment is red. And so there's really not a skin pigment, but it affects your skin color. Make sense? Yes, that's basically